Congratulations! You've made it to HIV Part 2, the viral life cycle. And you've been dropped right into the middle of an epic battle. So, as the battle unfolds, let's highlight the stages of the viral life cycle. Then head over to Sketchy Farm to learn about all the tools we have in our arsenal to thwart the White Wizard. First, the history of this conflict. HIV targets host CD4 plus immune cells, including macrophages and helper T cells, which are HIV's main target. Like other viruses, HIV takes over, i.e., commandeers, the cells it infects, turning them into virus factories, just as the White Wizard has seized the neighboring CD4 castle. From that neighboring castle, he's prepared a new offensive, generating new virions, which are complete mature viruses that are ready to invade the next cell, or in this case, new ships ready to invade the next castle. Before we jump into the life cycle of HIV, let's introduce three important structural genes: GAG, ENV, and POL. These genes are responsible for encoding the components of the virion and important viral enzymes. As this brand new invading virion ship heads for this new castle, notice it has prisoners on board in the center of the ship, gagged and tied around a 24-hour sundial. This is because the GAG structural gene encodes virion core proteins, like P17, the matrix protein, and P24, the capsid protein. You may recall from HIV Part 1 that P24 is an early antigen detected on 4th gen testing. That envoy boat is carrying plans for a major attack called the Big One, with siege towers and grappling hooks galore. Let this envoy represent ENV, a polyprotein containing the precursor envelope protein GP160, which is cleaved by the host cell into GP120 and GP41. GP120 and GP41 play an important role in cell entry, which we'll talk about in a bit. Enter Paul, the blacksmith, a blacksmith of enzymes. He's here representing the POL gene. Like ENV, POL encodes a polyprotein which is cleaved to form important enzymes required for viral replication, including reverse transcriptase spellbooks, integrase keys, and protease blades. We'll come back to these enzymes in a moment. Now that you're familiar with the three structural genes and the protein products they encode for, let's see how they each play into HIV's life cycle. The outer wall of the castle will represent our defending host cell membrane, and it looks like this battle is about to heat up. Entry into a host cell is mediated by the HIV's envelope glycoproteins. This starts with the attachment of GP120 to receptors on the host cell surface. GP120 binds to both the primary receptor, CD4, and the chemokine coreceptor, CCR5 or CXCR4. Up there on the castle wall, our brave Helper T Squire is under attack. The siege tower is binding to the castle wall and preparing to unload its contents. While the GP120 siege tower is for HIV attachment, the GP41 grappling hook is for HIV fusion. Once GP120 binds the host receptors, GP41 undergoes a conformational change that leads to fusion of the viral membrane, aka viral envelope, with the host cell membrane. Now we see the White Wizard's camp storming the castle, trying to fuse to the wall with these GP41 hooks. Upon entry, the virus uncoats, releasing viral RNA, enzymes, and structural proteins into the cytoplasm. Once the wizard gains entry to the castle, he gets to work, turning the castle into a factory for the creation of new viruses. The virus actually carried with it those POL-encoded enzymes, reverse transcriptase, integrase, and protease. And now that the virus is uncoded, the enzymes are free to use. First, HIV uses its reverse transcriptase to convert its single-stranded RNA genome into double-stranded DNA. And so, our retroviral wizard is using his reverso transcriptome spellbook to turn his single-file RNA army into a double-file DNA army. Next comes integrase, yet another product of Paul the blacksmith. This enzyme integrates the newly formed viral DNA into the host cell genome. Integrase will be represented by the keys the blacksmith produces to unlock the DNA tower, so that the wizard's double-stranded DNA army can infiltrate. Integration into host DNA is a smart move on the wizard's part. For one, it can't be undone. That host cell will be infected for the rest of its life. Plus, once the viral genome is incorporated into host DNA, the host cell machinery unwittingly transcribes and translates the viral genome alongside its own. Meanwhile, the ENV gene product, which is the precursor polyprotein GP160, gets glycosylated and then cleaved by host cell proteases into GP120 and GP41. This all happens in the endoplasmic reticulum and Golgi apparatus, because in eukaryotic cells, that's where membrane-associated proteins are processed. Remember, HIV is taking over the host cell machinery, so it doesn't need to reinvent the process. Notice how the reef around the ENV boat kind of looks like the ER in Golgi, the processing center for virus surface proteins. You should know that while the ENV polyprotein GP160 is processed by the host cell, the GAG and POL polyproteins will be cleaved by HIV's own protease. So these polyproteins, along with other HIV components, make their way to the cell membrane. The immature virus then buds off the host cell membrane, forming the viral envelope from the host lipid bilayer along with GP120 and GP41. So now we're done, right? Nope! Ha! Remember, the POL and GAG gene products are still in their precursor polyprotein form. It's not until the end of the budding process, or shortly thereafter, that these polyproteins are finally cleaved by the viral protease, which is the third and last POL gene product. With all the products cleaved and in their active form, we've finally arrived at a mature virion ready to infect new host cells, and the life cycle is complete. So far, we've watched HIV mount a highly successful offensive. Of course, the host does try to fight back, though the key word here is try. Part of the host response is the formation of neutralizing antibodies. The primary target of such antibodies is GP120, the outer envelope glycoprotein. Unfortunately, the HIV virus has a few ways to counteract this host antibody response. For one, GP120 displays a high degree of antigenic variation, which prevents the host from mounting an effective antibody response. In addition, dense glycosylation of GP120 shields its protein core from recognition by host immune cells and blocks the binding of neutralizing antibodies. As a result, the host is typically unable to form an effective neutralizing antibody response to the virus. Let's finish up by briefly mentioning treatment. All patients with HIV should be on antiretroviral therapy, or ART, to prevent the virus from wreaking havoc on their immune system. These drugs have been designed to target specific parts of the life cycle. You see, we knew there was a reason we needed a microsequel. For more detail on each of these drug classes, be sure to check out our antiretroviral sketchy farm sketches.